I am Mr. Wilson. Welcome to the first in our classes on sampling techniques. You can find us on the internet at tcp-academy.teachable.com, and we're on YouTube at CSEC Biology TCP. Today we're going to be looking at the use of a quadrat as a sampling techniques, and we're also going to be looking at observation as another sampling technique. It is impossible to count all organisms on Earth, and sampling techniques are employed to sample small areas. The data obtained is used to make decision. Might be conclusion, the data is analyzed, but one would appreciate the fact that it is impossible for us to count all organisms on the planet Earth. Hence, we have to use some method of estimation that can bring us as accurately as possible to our reality. Now, examples of techniques that we're going to be looking at in this course include observation, quadrat, line transit, belt transit. And we're also going to be looking at ways in which we could actually estimate population size. We're going to look at the mark and release technique. All those are going to be covered in this course. Today, we're going to be looking at two, observation, and of course, the use of a quadrat. For any ecological activity, the air to be sampled must be observed for adaptation that supports the existence of organisms in the area. The interrelationship and intra-relationship must be noted. Now, the interrelationship speak to the difference there is between organisms within the community, while the intra-relationship speaks to the differences there are between organisms within the population. So we understand population to be organisms of the same species and community to be pretty much different species. Now, this clearly indicates that good knowledge of biotic and abiotic factors will be needed for any observer of the environment. This will help the observer to, of course, obtain credible results are results that are both reliable and valid. So once you go out to do any ecological study, whether you are using a quadrat, a line transit, a belt transit, whatever you are using, you must always observe what is happening. We're sampling with a quadrat today for CXC biology, and we are going to be outlining a number of things that are supposed to be captured in your lab if you are to sample with the quadrat. If you are not sampling with the quadrat, once you go out to do some sampling, there are some stuff that should be captured in your assignment or your lab report. And we're going to be looking at those today. So let's find out what the quadrat actually is. The quadrat is an apparatus used to sample stationary or slow-moving organisms like plants and slow-moving animals, example, our millipedes. It's a 1 meter square shaped frame. However, smaller types can be found. It is made from metal or other suitable material. In my class, when I give the kids a project to like make a quadrat, they tend to make it from PVC pipe. How does the quadrat work? It is thrown randomly in the identified sample area, and individual organisms being sampled are counted at each row. The number of organisms is recorded in a table after each row for that row or a 0 for no organism. So if no organism was obtained, the organism that you're looking for, you record a 0 in the table. The number of throws should be about 5 to 10% of the sample area size. More throw will result in a greater accuracy of the outcome, Organisms that fall halfway in the quadrant or more are counted. So if it's less than halfway, you're not going to be counting that organism. Where organism cannot be distinguished like grass is growing pretty much like on a mat. The quadrat is made into smaller squares using string, and the percentage area covered is estimated. It's very important to note this bit because when we can't do accounting, we look at species cover, and this approach is the approach we're going to be using to obtain the result. It is used to aid the study of distribution and abundance of organism throughout a uniform ecosystem. Now, the having thrown here quadrat, there are a number of things that we could do in estimating population. Calculations like percentage frequency of that species, the density of a species, the species coverage could be looked at, population size are just some of the calculation that are usually done when we are using the quadrant sample. Now, all these we will have to learn to do and have a good appreciation of the formula to ensure that when we write up our lab report, we are doing an awesome job. What is required of an individual or a student carrying out a lab of this magnitude? First, they should carry out careful observation of ecosystem, document organisms observed. So you're going to need to master a table. So you must construct your table to record your observation. Identify symbiotic relationships in the area. Identify, in this case, two types of ecosystem found in the area. Construct two food chain / food web, one from each or one for each ecosystem using organism that you would have identified in the area. Cite the benefits derived by organisms from both ecosystems. Name and explain abiotic factors observed in relation to the organisms in the area. Identify pollutant in the area and explain the impact they have on the ecosystem. Outline ways in which pollution can be reduced. Identified the need for afforestation or reforestation in the area. So you will have to do a distinction there, which is needed and why. So all those are things that we're going to be looking for in this ecosystem that we're going to be studying. So if you see or observe on the screen, you're seeing that it is reminding you that you should observe the area. Now, with all those questions that we're supposed to answer, we're expected to look at biotic and abiotic factor. And you're looking there, you're seeing a windmill up there, you're seeing that it's a bright sunny day. These are things that you can observe from the screen here. And if you observe, there is somewhat a terrestrial ecosystem here, and you're seeing a river running down here, and we're seeing water, a water body here with some organism. We have to pay attention to that. You have to pay attention to all the organisms. Some are actually, you're seeing the picture, while others you might see in helping you to identify, you're seeing words of organism and sometimes pictures. And of course, you'll have to observe because you never know. There might be a bird flying across or something moving across the ecosystem that you need to look at. Look at organic waste, paper, plastic can, uh, lot of plastic here, just few plastic here, a little bit of plastic here. We are seeing some fabric here. You have to pay attention to that. Now look here, we're having another here. Here it's all brown and it suggests that mining is taking place in this area. We're seeing an animal here as well. So let us, having done that, let us look at what is expected or what's happening in the ecosystem, as it were, for sampling. So now that we have done our observation, we have recorded nicely in our table that which we need to record in order to fulfill the requirements of this lab. We are going to now move to the next step to identify the sample area that we're going to be working with today. If you observe, we have just put in two flags as these are marking the area for which we are going to be studying. And another flag, the flags are in. And then the other thing we're going to be doing here is to measure the sample area. So you would have observed the arrows there showing the sample area being measured. Pretty much rectangular area is identified for sampling. The other thing we're going to be doing, we are going to be throwing the quadrat randomly in the ecosystem. So that's the first throw. And when we throw the quadrat, we want to make sure that, of course, all organisms that are more than half that are in the quadrat, we are going to be counting them. So if you observe this organism, there's a part of the plant on the outside, more than half is on the inside, so it's counted. So we are counting a particular plant here. We'll soon look at that. So all these plants, we're not going to be looking at all the plants here. There's, of course, a color plant that we're looking at, be it white, yellow, blue, or pink. We must identify the plant that we're studying or the organism that we're studying before we start showing that quadrat. So we move again. You could just choose a plant there that you want to study and, of course, see how many of those plants we are getting in the quadrat. Now, having done that, the next thing we are going to be doing, we are going to be recording our observation. So for the first quadrant, let's go back up. For the first quadrant, if you observed here, we have 4 yellow plants, 1, 2, 3, 4, 2 blue plants, 1, 2 white plants, 1 pink plant. Now, if you look at the quadrant, it is saying individual counted. For the first row, it's 1. So this clearly tells us that we are looking at the pink plant, as it were, for this row. So this is just showing you that we're going to be throwing this quadrant 10 times in the sample area. And for each row, we are going to record the number of organisms in the cells here. For each row, individual counted, we are going to record it here. So we really don't want to wait until the end of the activity before we do the recording. We might just forget something. So we go again and we throw randomly. That was just an example there. And this is a random throw again. And we move right along. Another random throw. So that's 3 throws. So we would have been recording whichever organism we are looking at. Then we go again. That's another random throw. Another random throw. Random throw. Another random throw. Now look at this particular throw. You will observe that the quadrat is going right through these 2 organisms, right in the middle. So of course, these 2 organisms will be counted among the organism if we were supposed to be looking at this particular organism. This would be counted as part of the organism in the quadrat. Right? So remember, if it is half or more than half, we're going to be capturing that. Now, if we observe this, here we come upon another, we have to make the decision whether this is half in or half out. All right. So we move on, quadrant, and that would have completed the throw. And there we are observing some birds moving across. So again, we have to document in our table that we have seen birds, and we want to see if we could identify the type of birds that are moving across, because there are many birds moving across, and there's this big bird going across. Is the arc? I'm not too sure. Probably you could tell me, as we're expected to do a food web or a food chain. So we need to have a good appreciation as to what these birds are and their role or niche in the ecosystem. I've seen some human moving there on a part. I don't know if it's an exercise part, but they keep coming and going on that part, which suggests that that's what's happening. Here we have a table to populate, and it refers to, it looks at the organisms that we would have observed in the area, pollutants that we would have observed in the area, possible relationship that we would have seen in the area, be it parasitic relationship, be it a mutualistic relationship, commensal relationship, we want to make sure that we are seeing that. And if there's any other thing that you would have observed in the area, probably you want to talk about abiotic factor or anything like that, you could pretty much record it in your table there. So it's just a matter of practicing to record in tables. It makes the work much easier. When here you want to construct your food chain or food web, please be reminded that a food chain or food web, they all begin with a plant or plant-like species. All right, it does not begin with the sun. Distribution of organism. The distribution of organism is influenced by biotic and abiotic factors. Biotic factor refers to living interaction, which would include listed there disease, predation, symbiosis, competition, pollination, camouflage. And then we have abiotic factors, which are the non-living component or the physical factors. And these include light intensity, rainfall, wind speed, temperature, water, soil pH, water salinity. And we're going to be covering this more when we cover abiotic factors as a topic. Now here we have our table for the data that we were collecting, and you would have observed here that all the cells are populated. So name of organism sampled, the individual, which organism was sampled. You want to have that name of the organism being studied. You want to ensure that you write it down. In this case, it will be the color of the organism. So you could just tell us which organism it was in the comment below. Of course, we were able to pretty much record this organism 22 times. And now we're going to be looking at ways in which we can estimate the population. Now we want to find the total number of species found. Now this is a sum of the individuals. So all that we did was to pretty much add all these here. The sum of individual, 22 of a said species were identified. All we did was to add it up. Now the area of sample space, we want to find that. What's the area of the space sampled? Remember when we did our measurement, it was 40 meters by 20 meters, so we have 800 meters square. Very important for us to do all these calculations. Number of individual per quadrant, one might say the mean, average. So that's going to be 22, the 22 individual divided by 10, which is going to give us 2.2. And that is pretty much an average or mean individual per quadrant. Then we move on down to the density. How do we calculate density? It's the total of a species divided by the number of throw for a 1 meter quadrant. So the density here is pretty much the same as the mean, which equals to 2.2 individual per meter square. Now it's very important to note that you must be able to transpose these formula because there are cases where you are given just the answer and probably you are given the number of throws and you are supposed to find the total number of the species or you are given the total number of species. You are supposed to find out how much throw it was. So you must be able to transpose the formula. You could be given this anyway to find your answer. The population size is equal to the density multiplied by the area of land. So the area of land we had was 800 meters square and the density was 2.2. So we multiply that, we get 1,760 individual. Now percentage frequency is the number of quadrat in which the species was found. So when we threw those quadrat, if we threw it 10 times and we found the same organism in all 10 throw, then the frequency there would have been 100%. But suppose we threw it 10 times and we only found the organism 5 times, then the frequency there would have been 50%. Right? So to find your frequency, it is the number of quadrat in which the species is found divided by the total number of quadrat. And then you are going to be multiplying that by 100. That gives you the percentage frequency. Now, species cover is, of course, the total percentage cover divided by the number of quadrant row. And this is pretty much used in area where the percentage of quadrant cover was estimated. You can't count as if you were using a graph. So you divide your quadrant into smaller frames using strings, and then you estimate, or you estimated, the coverage of the area. In that case, you are going to be adding all 10 percentage cover for the 10 throw you made, and then you can give your percentage species cover for the particular area. Now, it's very important that when we carry out an experiment, we can reach a conclusion having done our observation and analysis. Now, here, to record that conclusion, it could be done in a table, and your conclusion is going to be based on what you set out to find in your aim. You could record it in a table, you could record it by writing in prose, like a sentence, a paragraph, or you could use a bar chart if that is pretty much relevant to your study to report your findings. That takes us to the end of how to use the quadrat and use the data to estimate outcome or do calculation. We would have done all that. Please feel free to replay the video however many times you so desire to ensure that you understand. And two, identify a species using the quadrat by color or shape and throw and do your calculation and tell me what your result was. I am Mr. Wilson from TCP Academy, tcp-academy.teachable.com. You can find us on YouTube at CSEC Biology TCP. Find our website, it's right there, tcp-academy.teachable.com. Sampling techniques produced by TCP.